Science has always been a passion of mine. From early on, I knew what I wanted to do. I wanted to be a scientist. From probably junior high biology classes on into high school biology classes. In fact, I remember one day sitting in a 10th grade biology class and thinking, I want to do this for a living. I think I can do this for a living. I want to do this for a living. And really from that moment on, I have been on a path to be a professional scientist. When I was a graduate student at Emory University in Atlanta in the mid-90s, I was working on a project that involved how cells signal to each other and some of the cell behaviors that resulted from that cell-self signaling. And as part of that research, I discovered a protein called SCAR. And it turns out, I didn't know what SCAR was at first, but we began to get deeper and deeper into this. We realized that this was a key protein for understanding how cells crawl around. And so, after I finished my PhD, I went off to MIT in Boston and started my postdoc in the lab of a guy named Frank Gertler. He was just setting up his lab, and in that lab, I was given the opportunity of setting up a new microscope for him. And his lab studied, again, how cells crawled around and some of the cytoskeletal elements inside of them. And I got the microscope set up and I made my first movie, and it was just a revelation. I got so excited when I was able to make a movie of a cell crawling around that I just fell in love with the problem. It just became fascinating to me how this worked. Coronins are a fascinating family of proteins. They've not been widely studied, but they actually are very old. So we think that coronins have been with us for nearly 1 billion years on planet Earth. These are proteins that seem to develop when we moved from single-cell organisms to more multi-cell, complicated organisms. They've been with us the whole time. And to me, something that's that old is doing something interesting, even if we don't fully understand it. That was really one of the bases on which I founded my lab. And so what we have done over the last 6 years is really try and take apart this family of proteins, figure out what that does. We've known about what the part is there, but now we want to understand how it works. And so one of the ways in which we try to understand how it works is take it out. And when we take it out, we see what the system can do without it. It's like essentially trying to figure out how a car works by taking out one part at a time. Maybe not the most efficient process, but frankly, really all we've got at this point. With that, along with imaging that we do here with the microscope, we're able to dissect the basic function of this protein. What we have discovered is that this family of proteins, the coronins, help to break up that actin network and to help recycle the components and essentially just kind of lubricate the entire machine. And one of the repeating protein domains that coronin is made up of is a domain called the WD40 domain. And the name is a little bit obscure, but it was sort of a cute name. The irony comes in the fact that we have discovered that coronin really does loosen up the actin cytoskeleton. So it's, not necessarily something we planned, but one of those happy serendipities. In addition to understanding this basic mechanism of how coronins work, we're actually also directly working on melanoma and trying to understand some of the proteins that are changed when that disease really gets going. It's a cancer where metastasis is centrally important. We don't have a lot of good therapeutic options. And for me personally, my father died of melanoma in 1993. When I was a young graduate student, he fought a battle with melanoma for several years, and eventually it took his life. And at the time I thought, I want to do something to try and make it so that other people don't have to experience this. 50 of us were selected out of this original group of 2,100. And again, I feel incredibly lucky to be included in that group. What it is a 6-year appointment with them that supports both my salary and provides research support for the lab and some equipment purchases. And it gives me access to some of the HHMI, or Howard Hughes Medical Institute's resources, but I'm also invited to go to meetings and to meet with other Hughes-funded scientists. I hope that we can have a complete understanding of how coronin works. We've gotten a good start on that, but there's a number of important issues that still need to be sorted out. That's one aspect of what we want to do. Another really important aspect is we want to push further with this melanoma model. We feel like this gives us a really unique take on the disease state. And so what we're doing now is we're beginning to pull in people from the cancer genetics field. We want to understand mutations that really happen in patients, begin to incorporate those into the model, and see how it affects the process of this cancer metastasis or spread. So that's another major part of the lab. A third part of the lab is, frankly, a little surprising. I'm getting more involved with some chemists. And I'm a biologist, and chemists and biologists are kind of like cats and dogs. But it's becoming clear to me and many other people that science going forward is really going to require a multidisciplinary approach. We can't just keep doing the same experiments and expect to sort of make breakthroughs. We need to start talking to chemists and physicists and engineers so that we can begin to really design and develop new approaches to be able to probe the biological problems to a greater extent. You should follow your passion. That sounds a bit hackneyed and it sounds like generic advice, but it really is true. I do what I do because I love it. I can't think of a better job I could have in this world than